Our speaker at the moment is Petra Gutenbrunner, and then she is a PhD student at the Max Planck Institute of Biochemistry in the Jürgen Koch's group. She worked as a software developer for about 6 years, and then she studied biomedical informatics at the University of Applied Sciences, Upper Austria. And then she received her bachelor degree in 2014 and the bachelor degree in 2016. In the meanwhile, she also did some internships at the Wellcome Trust Sanger Institute in Cambridge, and then she moved to work with Jürgen Koch in 2016. Now she's going to tell us about how to configure Andromeda scoring function.
I'm sorry, the plugs are not working. I don't know if it's for other rooms.
The plugs?
Oh. Yeah, I will ask them, but yes.
So yeah?
We can start.
Yeah, sure. Peli? Sorry, Peli. Sorry, go to Peli.
But I guess we can all start now, right? Even though the plugs are not working at the moment, we will fix it, but... Okay, okay, perfect.
Yeah. So, we're just quickly changing the batteries for the presenter. So, welcome everyone. Can you hear me? Yeah, I think everyone can hear me, right? I hope so. So I hope you had all a good lunchtime and we can fully energized start now to the first tutorial session, which will be about the configuration of the Andromeda search engine. And so we have already heard a lot about modifications and Pavel? And how to configure new things, and we will learn now how to do this. So also, I will show everything here on the screen. You're very welcome to follow me. And if there is anything too fast, we also, and you need additional information, we have also some helpers here, which you also can ask anytime. Good. And of course, you can also ask me. Sorry. So before we get started here, you've all got an USB stick, and maybe you have done that already, but I would suggest please copy now all the data from the USB stick to your laptop, because the data you will need now in this tutorial and also in the following tutorials today and the following days. As next, so we're talking here about Andromeda. So today we are learn how to configure new parameters. So like there was already a question today, there is a modification not provided in MaxQuant, how we can add this modification. So in this session we learn how to do this. And also tomorrow will then be a lecture about the search engine itself and how it does the peptide identification. And if you can't wait until tomorrow and you want to know everything already today, there is also a very nice paper about the Andromeda search engine, and you can already read it up. So what we are going to do now is that we want to add a new modification. And in general, I also want to mention there is already a lot modifications and so on pre-configured in MaxQuant. And so for most of the experiments, you don't need to do anything, but there are, of course, specific experiments where you need specific information, and therefore it's good to know how to add all this information that you can customize this. So we add a new modification, we get the information from a well-annotated database from Unimod, As next, also we heard a lot about SILAC labels, and now we finally see how to add new SILAC labels in MaxQuant, and this is then important for the quantification. As next, we learn to configure a new protease. So we've also heard during the sample preparation lecture of Tammy that sometimes trypsin doesn't work and you need a specific protease in order to identify peptides. So here you also learn how to do this. And the final step is then the configuration of a new protein database. So let's start with the first assignment, which is where we add a new modification. As an example, we just picked the dehydroalanine from tyrosine. As I mentioned, Unimod is a very good address to find all this information for this additional modification. And so just a brief view how the information looks on Unimod. So yes, you can see here the name, but I will also explain it in detail. So you have then tyrosine, then the modification, then you end up with dehydroalanine. So I would suggest we go directly now to Unimod to get this information. So here you go then to the browser, and we type in here unimod.org. We can't see it here. Usually when you go the first time to this web page, there is also a login screen. And you don't need to log in here. You can also type sign up as a guest, and then you also come to this page here. So here you have a nice overview of all possible modifications, and then we also have here some filter fields, and there we look for the modification of interest first. So we type here the dehydroalanine, and then we can click here search, and then the results are filtered here. So we already have some informations here, but for our case, to add this new modification in MaxQuant, we want to have additional information, so we click here on the very left on view, and then we come here to this window which I've already shown you. So let's go quick through the most important fields. We have here the description of the modification, then you can see the composition of the modification, and then also the monoisotopic mass, and as next we can see here the site, so the amino acid where the modification can occur, and then also information, sorry. Yeah. Is that better? Yeah. Then we have also the position here. So is it on the C or N term? And yes. So we know now all the information in order to add a new modification. So let's go back. And yes, so now we are at the point where we start MaxQuant, and I think that's the first time this week for some not. And so let's do this. So when you go to a USB stick, and there you have find a folder software, there is also MaxQuant. So you go in this folder, and then you have the exe file here. And so we start MaxQuant. Yeah. So maybe a question, how many of you are using MaxQuant the first time? Ah, nice. And how many of you are already experienced, or yeah? Okay. Also very nice. And do we have any expertise also here? Oh. Yes, so at the end we are all hopefully expertise, right? Good. So for the ones who are using it the first time, I quickly explain the structure of this. So we see here the GUI, and it's separated in a few tabs. We can see here at the top. So the first tabs are raw files. So if you do an analysis, you can define your experimental files here, the raw files, and which you want to analyze. But also, I just go briefly through it, and in the next sessions, they will be explained in more detail, so don't worry. As next, you can, when you define your raw files, you can also group them. And when you have more groups, in the next tab, in the group-specific parameters tab, you can assign specific parameters for each group. But if there are parameters which are not specific for any group, you can define them in global parameters. And in the next tab, you see in the performance tab, so when you start an analysis, this gives you an information where this analysis is at the moment, so at which step. And of course, here's also the important button to start the analysis. And you can also define a number of threads for the multiprocessing, that it's a bit faster then. So then we come to the viewer. The viewer will be explained by Jürgen in the following session. And finally, we are now in the configuration tab, and here we will be for the rest of the tutorial. So this is the one for us. So the first assignment was to add a new modification, and we can here see that there is also a tab for this where we can click. And as I mentioned, there is already a lot there are already a lot of modifications pre-configured. And so when you want to add a new modification, it's also useful to find if the modification is already present or not, because then you don't need to add one when it's already there. But in our case, we know I know it's not there, so we add a new one, and this we can do with these buttons here. And on the right side, there is then a window appearing, and here you add all your information about this modification. So maybe it's a bit overwhelming, so you might don't know where to put what. So a quick tip would be to go check a modification which is already defined, to know where to add which information. And I guess all of you know phosphorylation, and we can also click with Ctrl F to look for it, and then we say phospho, and then we find next, and then so that's also a nice way to find which information is already present. So here we see phospho modification, and on this example I will explain you all these fields. So at first we see the name of the modification. This is an important field, because when you then actually configure the parameters in order to run the search, and there you also have to select modifications, you find this modification based on this name here. And was there a question or no? Okay. So the next is the description field. This is arbitrary, so that's important for you when you define a new modification, because maybe you need something to distinguish, and that you can define here. As next, we define here the composition, and then the position, if the modification occurs C term, N term, or it can be anywhere. And then also the type is also a very important field, because so in case when you add a new modification, you have to select here standard for standard modification, and we will come to that point later when we add a new label, but you can also select here different values, like a label when you want to add this, and this information here is important for MaxQuant and Andromeda in order to be able to interpret this entry. If the modification creates a new terminus, you can also select it here. And as next, you can select the specificities, like on which, yes?
So in the
Substitution is, for instance, when amino acid is substituted. So also maybe an hint when you don't know what the value can mean. There is already there are many entries, and you can also check here, like there is a substitution entry, and then you can see there is a replacement, and also this changes then the mass, and therefore Andromeda needs also to know this in order to find this. Okay, then to specificities. Oh, yes. So here you can define the amino acids where the modification can occur, and in the phosphorylation here is on serine, threonine, or tyrosine. And also you can define here neutral losses or diagnostic peaks. Okay, I've explained now hopefully everything here. And let's create now our new modification, the dihydroalanine. So before we added already a new entry with add. This appears at the bottom of the table. We click on that, and then we have here the new entry, which we will modify now. So we looked up the information on Unimod, and I'm just lazy and go there and copy all the information which I want to use. And so here I type it in. I make it a bit shorter because you select this then when you configure the search. So I just say here tyrosine. Yes. And as next we set here the description. And for the composition, by default, there is always water. So to define this for the new modification, you delete this entry, and then you click here on change. Yes, you have two options here how to define the composition. Either you click here on this button, then you get the table here, and you can select here the elements you need. And as next, we also offer a combo box where you can directly select the element. Okay, so let's go back to look which composition we need for this modification. Here we have hydrogen, which is reduced by 6, then carbon also reduced by 6, and oxygen reduced by 1, subtracted, and then we go back here, and then I've already selected hydrogen, and with these arrows here you can add them and subtract them, like here. And here we need -6, so we select this here. And as next we have 6 carbons, which we subtract. And then we subtract 1 oxygen. So you might have also seen that the mass value here is changing. So depending on the composition which you define, the monoisotopic mass is automatically, yes, calculated?
I'm sorry, from this row here, I can't see anything. Is there any way how you can zoom in?
Yes, I don't know if we have a chance to do this. Yeah, is it somehow possible to do this? Can you try? Just click OK here, please.
This. I have to go to desktop.
Here.
This is this. Like, let's try medium. I have to log off.
No, that's not good.
Okay. And let's try the resolution. Where is the... Let's get rid of this throw quota thing as well.
I mean,
No? Just 1 second. Before storage. And this is... There we go. Better?
is it better now?
Sorry,
Yeah?
sorry, we have a question. Yeah.
Okay.
Yeah, well. No, yeah.
So? So everything fine or better? Yeah? Nice. So we were at the point where we wanted to define a new composition. And we have done this here. Just go here. And based on the composition which we select, there is the monoisotopic mass automatically calculated. So we also have the monoisotopic mass on the Unimod web page, so we can also go there and check if it's the same value to check if the calculation is correct. And it's minus 94.041. So go back. Yes, it's the same, so that's good to know. As next, we want to define the position, and this is also the information given at Unimod, so we can see here it's anywhere, so we can leave here the default value, and the next value is important, so in terms of modifications, we leave standard here. And it doesn't create a new terminus, and by default here, where you define the amino acids, it's lysine, and we remove this with clicking the minus here, and then we add the amino acid, and here we can select tyrosine, because the dehydroanal is happening here, sorry. Yes, and in order to add this modification, we have done now everything. So the next step is also very important, because I'm not doing this here, but if I would select another entry in the table, all this information would be updated, because it will show the values of the new entry. So before you do that, and then you have to click Modify Table in order to keep the changes, because here you still see the entry has new modifications, and when we click Modify Table here, the table is updated. So that's a very important step. But in order that Andromeda knows that the value is there or changed, you also have to click Save Changes, because then it's written to a database, and Andromeda can read this then, or MaxQuant. Let's go back to the presentation. So we have finished now the first assignment. Are there any questions regarding to this assignment? Yes?
How to add, for instance, hexones, so light reservations? Because then you find in the list of the elements.
And did you find the Unimod information for?
Yes, for like hydroxystrobin.
So, Pavel?
Yes.
So I don't know. I haven't. So I don't know. Is there time then in the bootcamp to have add the specific values? That's fine. So can we check these detailed questions, how to add specific one in the end, if that is fine by you? Yeah. Okay. So let's go now to the second assignment, the definition of a new SILAC label. So in our case, we want to add here a new histidine 8, which is a heavy histidine. It's similar to the heavy lysine or arginine. And we have also here the composition information. So we have 5 heavy carbons and 3 heavy nitrogens. So it's roughly a mass change of 8 Daltons. And we go back to MaxQuant again and to add this new SILAC label. To do this, we're still here in the modifications tab. And so as I said, it's similar to the labels which are already predefined, so we can also have a look on them, how they are configured. And then in MaxQuant, there is also a nice feature, which is here. We can duplicate an entry. So if we do this here, we have a new entry, which is the copy of the previous one, and this we can then modify. So in our case, we can change then these values to histidine 8. And this is also a nice way if you keep adding similar modifications, because then they have all the definition has then all the same structure. So here we modify this. We said these are 5 heavy carbons and 3. So this is arbitrary, this information. It's just good to know for you. And it's on the histidine. As next, we change the composition. So we remove the values here, and then we click change. Here, in order to set the heavy isotopes, we have to remove the light ones. So here we know we have 5 heavy carbons. We remove now 5 light carbons. So we do this, we select the carbon, then we reduce this, and then we have to add the heavy version of the carbons. And this we can do when we go there, and then it's carbon followed by an X. And this gives you the information that this is now the heavy version, or the heavy counterpart, and here we add 5 of them. We do the same with the nitrogens, so we subtract the light ones, and then we add the heavy ones. As I said before, we should have a mass of 8 Daltons, and so this we have here, so the composition is correct. When we click OK, we see here these changes, and as next, again, we have here the type column, and because we duplicated the entry before, we have already labeled, and this is also the value we need. And we can leave this here. And the rest, so we need to change the amino acid, so we remove that and select histidine, because we're talking here about the histidine. So now we have done everything in order to add a new SILAC label. And then we have, as usual, what is important to click now. Exactly, and right, very nice. Okay, so let's go back and see what we have to do next. So the next is I want to draw your attention to the dependent peptides. So when you define modifications in the search, in order that the search engine looks for the additional modification, you're very biased. So if you would not set that, it would be then not possible to have this peptides with this modification in the result. But here there is a very nice feature, which is dependent peptide, and it's also more unbiased. So it looks for other modification which can be in your experiment, in your sample, and then it gives you a suggestion, and then you can look. So. But this is then explained in more detail by Jan in the lecture tomorrow. As next, we want to configure a new protease. So we go again back to MaxQuant, and we are still in the configuration tab here, and we change now the first time this view here. So we click here on proteases to add to manage them. So for the most shotgun experiment, we have already the protease defined, but of course there can be an additional one, and we learn how to add a new one. But before I said, let's have a look on already predefined protease. So everyone, I guess, knows trypsin. So let's see how trypsin is defined. It's not a lot what you have to do here. So first you add the name, then you have and this is also when you configure then the search engine, you select the protease based on this value, and then you have the additional information. And as next, you can see here a matrix where you can define then the cleavage rules. So let's add now a new protease. For this, we click here Add. Okay. Then we have here again a new entry which we can modify. And for instance, we want to add here a cleavage rule when there is an alanine, and then it should cleave. So alanine and cleave. Then we can also here see like how the descriptions look for other ones, and we can also take this suggestion. So cleaves after alanine. And now we want to, yes? It's not activated, so you can't click on add or? So there is a hidden button. Maybe you can click discard changes. So if it's locked and you can't press any buttons here, then when you click on this arrow here, maybe you click discard changes and then it's possible. Does it work? No? Yes? Nice, good. Okay, now we want to define here the cleavage rule. And here we know when there is an alanine, then a cleavage is happening here. So we can, there are two buttons, and this is very nice because you don't have to select each value here, you just click here plus. Yeah, it's a little bit small here. And you can also unselect everything at once. So, and we also want to define an exception here. So if there is an alanine followed by a proline, then it doesn't cleave. So we deselect this, and that's all. So here you can easily define your rules based on the enzyme. So as next, as we know, we click Modify Table, Save Changes, and we have now our new protease. Opala. Yeah.
Perhaps I missed it, but how do you define whether it should cut on a C or an N terminus?
This is not at this position because then later in the search, when you define it, then you have additional information as far as I know, where you say the specificity and then it's a specific enzyme or not, and this you can configure then when you actually define the parameters for the search.
Thank you.
So are there any other questions? Or yeah, okay. So now we are at the point where I think it's also the last assignment where we want to configure a new protein database. So this is the case if you are interested in a specific species and which is not already supported in MaxQuant. So in our case, we want to know, we want to analyze the kangaroo rat, and therefore we want to configure the kangaroo rat database. And so first of all, we need the database itself. And on the internet, there are two very well annotated databases, the Ensemble and the Unimode, where you can take your FASTA file from. And so let's go to Ensemble. and have a look how we can download the FASTA file.
But now you're kind of using A like this. Oh, wait. So
In
the second position
Pavel's
you
lecture,
need
we
to,
already
like,
have
to
seen
next to define
Ensemble,
Z.
and
So
he
you
also
like
downloaded a file. But here,
strange for
when you
the
go
person, that's
directly to Ensemble, then you have here in the menu at the top, Downloads. There you click on it. And then here on the very right, you see Download Databases. You click also on that. And then we have the...
Okay, I will insert my password.
Oh, amazing. Okay, so I'm lucky here it's still working.
But just
So,
something, okay,
and I'm already at the point where we see the
I will insert my credentials.
important information. So, when we are here, we can scroll a bit down and then we see the databases of all species, and then we filter for the species of interest. So I just quickly search for rat here, and then the table is here filtered, and then we see here the kangaroo rat entry. So here you can see its protein sequence, and this we want to download. So we click here on protein sequence FASTA download. And as next, we are redirected to the FTP server, and we can see here two possible FASTA files to download. One is the ab initio FASTA file, and the second one is the all peptides FASTA file. And for a standard experiment, I would say you would need the all peptides FASTA file, and it also contains the isoforms. But if you're interested in a protein prediction, then you would go for the ab initio FASTA file. So I have downloaded this FASTA file already, and yeah, so I go. Where are you? Sorry. At which point are you? Sorry. Which one? The protein sequence FASTA. Okay, now let's go to MaxQuant in order to add this new configuration. We are still here in the configuration tab, and then we click here on sequence database. And there are many databases already pre-configured, and so let's add a new one. This will take a bit because it has many things to load. Okay, so as usual, we click here Add, and then we get a new entry which we can modify. And here we can select then the FASTA file which we have downloaded, and therefore yes. So I select this FASTA file here and click Open, and then the name of the FASTA file will appear here. And please, if you select the FASTA file, don't modify this name, because this is important for Andromeda when you select then, the FASTA file for the search configuration, that for the FASTA file, the configuration is found. And this is kind of the idea in order to map the configuration to the FASTA file itself. The next text field is source. This is for you to define from where you've downloaded the database. So in our case, we write here ensemble. And as next, you can also define the taxonomy of this database. So if you have downloaded, yes, you need to uncompress this, yeah. That you have just the plain FASTA file and not the zipped version. So here I can, I explain, so here we can type either the name of the species, so in our case it's can. And when we have select this, we have here these arrows. So automatically the taxonomy ID is, we can have this when, yes?
The minutes, so people who haven't unzipped
can unzip. Has everyone unzipped the FASTA file? Okay, so just wait a second so everyone can do it.
Okay.
Can you do it with 7-zip or something like that?
Yes, but maybe
You have to me. So nice. I'm sorry.
yes, you can either
So,
here
you're
define
trying
the
to
name,
load database
Eurored, and then the taxonomy ID is figured out based on the name or the other way around. But I can. Yes, it's fine. So
Okay, it depends which one. Should I pick the mouse one? I like Arabidopsis. Okay. Whatever, it's kind of just bootcamp, you know, just to show people.
maybe you can later try this out with the FASTA file, and I would say we keep going here. Otherwise, we run maybe in time problems, if that is okay by you.
I'll show you example. So take zebrafish.
And of course, yes.
But I think you should.
Everyone likes it when.
No, we should go back.
Oh,
Have all.
the protein. Sorry, yeah.
Because the ab initio FASTA file would have additional information like 6-frame translation for gene prediction, protein prediction, and for the most standard experiments you would not need this additional. But I mean, of course, you can edit, but yes, you won't need it. Okay, so when we define the taxonomy of this database, we can either type in here the description and figure out the taxonomy ID. But if you know just the taxonomy ID, you can also type in the ID and then click on this arrow to figure out the description. So it works vice versa. As next, we are here. We have here the parsing rules, which we can define. This is important because the FASTA file contains many proteins, and you have to uniquely identify each protein. And in order to do this, you can define here several parsing rules. This is based on regular expressions, which gives you the opportunity when you have a string that you can extract certain information out of this. But there are already many regular expressions predefined for you, also for the most used databases, so you just need to use them. And later I will show you also a link, so if you have specific needs, you can also look up on a web page to learn how to define these regular expressions. So in our case, we click here, then select. So we want to extract out of the header in the FASTA file the identifier. And so here, if it would be the FASTA file from UniProt, we could select the first entry. And if it's from Ensemble, which is our case, we select this entry. And then we click here on X. We can see here then the identifier pass rule. Yes?
How would you know that's the correct one? Like Uniprot's obvious.
Yeah, we come to the point. I will show you later. So is next then also maybe the identifier then in the results doesn't, the ID tells you enough, so you want to have also the description of the protein. For this you can also extract the description information out of the header. Therefore you also click select, and in most cases you only need this one, and so we select this as well. And if the taxonomy information is also given in the header, you could also extract this information, and this is also helpful if you have maybe a combined FASTA file where you have the human proteins and mouse proteins, and so you can distinguish then these proteins. So here we can also define this one. Okay, and now to your question. How do we know if these regular expressions are working? For this we have the button Test Rules, which we click now here, and there we can also select here the FASTA file on which we want to apply these rules. So, and here you can see that we can see something, and that is already good. And so in the first column, we have the full header information. We can't see this all, but in the second column, we can see here the sequence information. So it's possible to get the sequence for each protein. That is also good. And now to these definitions of the parsing rules, here we can see a unique identifier. So based on the parsing rules, the header, so just this information is extracted. And then the same also for the description. And as far as I know, in this FASTA file, there is no taxonomy ID present, so that's why we don't see here anything. Okay, so as we all know, we have to click now Modify Table and Save Changes, and now we have configured everything. As next, we want to know, so as I promised, if you want to add specific regular expressions, you can go on this web page to look it up how to do that. And Jürgen mentioned also that when you have a database, also for the FDR calculation, it adds the reverse sequences of this. And also it's good when you add also known contaminants to this database, and these two things are automatically done by MaxQuant, which is really nice, because then you don't need to worry about this. And yes. So to summarize what we have done here.
Sorry, one question. Where can you find this contamination FASTA file?
Yeah. So for this, we have to go to the software where the MaxQuant exe is. And here you have a bin folder. And in this bin folder, you also have the configuration folder. And here you can see the contaminants FASTA file. We can also open this. And yes, so here the contaminants are defined, and if you have, because you want to add specific contaminants, you can also do this here.
We have a question.
Okay.
Is that contaminants file automatically added, or do we need to go through this process that we just did with that contaminants file?
No. This is done every
Okay.
time when you start the search. This is automatically added.
Okay, and if you wanted to add additional contaminants, you'd add it to that text file that you just showed?
Yes.
And it would automatically be used by MaxQuant?
Exactly.
Okay, thank you.
So to summarize it, what we have done here, we've added a new modification, a new SILAC label, a new protease, and also we configured a new database entry. And now the big question is where we do find this in MaxQuant.
Can I modify an existing database?
Yeah, for sure.
So-
I mean,
Let's go now to
Maybe I'll ask
MaxQuant.
but it's kind of, I mean,
And-
it doesn't make sense. You want to modify FASTA file, you want to...
If you go there now, all what we have configured now, this needs to be loaded by the software, because therefore we close now
Yeah, you
MaxQuant.
just should take your FASTA file and add this tag, like after or in the front end.
Let's say
And we start it again.
my database here.
Yeah, and you should kind of open this FASTA file, which
Okay.
you should load this FASTA file,
Okay.
protein, add a tag.
So let's now look up the first assignment where we added a new modification. Where do we find this new modification now? For this, we can go to group-specific parameters. Yes?
Just a quick question. Is the restart mandatory or?
Yes.
So, okay, good.
Because when you restart it, all the parameters are loaded and based on the definitions in the files, and so you have to do that. So it doesn't do it on the fly. No, also you store, with saving, you store it to the database, and then that really the software can read these entries, you have to restart it again. Okay, now we want to know where the modification is. For this, we go to group-specific parameters, modification, and we added here the dihydroalanine on the tyrosine. So voila, it's there, so all good. And you could also configure this for your search as variable modification, and yes. So we also defined a new SILAC label. For this, we go here to type, also in the group specific parameters tab, and this is where you define your SILAC labels, and here we have our new histidine 8 label. So that's where you can find it. And the third assignment was to add a new protease. For this, we go here to digestion, and here you can select the enzymes which you are using, and here you can find the new alanine cleavage protease. And there was a question at the beginning where you can specify is it on C or N term. This you would then select here. Okay. So the last assignment was to configure a new protein database. For this, we go to global parameters, and here we can add the FASTA file directly. And I also mentioned when you configure the database that the file name should not be changed, so this should be the same as here for the file which you select. So we are done now. Are any questions? Yes?
Is there any significant difference between the FASTA files downloaded from Ensemble or UniProt, or we can use either of them?
Pardon?
If there is any significant difference between the FASTA
You can
files?
use both.
Yeah, it's the same, right?
Yes, I mean, depending also on the experiment. So on UniProt, you can also define, like, you want to have just like, they're reviewed,
yeah.
unreviewed, and you have just a better option to customize or to define your FASTA files.
Yeah, but other than that, they are the same.
Yeah, I think they are also synchronized, so this should be also, the databases are also synchronized, and so the content should be also the same after a while. Are there any other questions, or?
Cannot comment on it. Yeah, the mic. Cannot comment on it. Like, did you compare UniProt with Ensemble? Maybe you can.
Yeah, seems like. Or maybe the.
Okay, well, no, we have one more question.
So yes, you're totally right. There's lots of synchronization. The only thing that you might notice is that some pseudogene sequences, so the sequences which are containing the asterisks in the sequence, in Ensemble, we don't import them. So that's the only main difference. No pseudogenes in UniProt, but for the rest, they are synchronized, so that they are pretty much the same.
Okay, thank you. Good.
Okay, thanks once again to Petra for the tutorial.